Hello everyone. So I hope my video is clear and I am audible.
Yes.
Yeah. Hello Emma.
Hello.
Yeah. So one of the things I would like to get out there straight off is that this is going to be an interactive session. So because the number of participants are not a large number yet, I would like everyone who can to turn on their videos if possible. So we have close to 10 people here and it is 4 o'clock already. So let us start. I hope the PowerPoint is visible.
Yes, it is visible.
Yeah. So let me formally welcome you to the PMRF-NPTEL session for the course Introduction to Developmental Biology. The PMRF, first let me introduce what the PMRF-NPTEL sessions are. So during the course of NPTEL courses, you will understand that there is a certain lack of interaction between the course team members and the students who are taking the courses themselves. The PMRF-NPTEL initiative is regarding just this. These sessions are problem-solving sessions in which a Prime Minister research fellow such as myself will discuss various problems from the previous year's assignments regarding the week that is currently going on. So current week that is going on is week number 1. So we are going to go through some sample problems of week number 1 from the previous year. And while going through that, we'll understand the various concepts and thus reinforce and have an interaction amongst ourselves of how to exactly approach the course as well as the problems. Yeah, I hope everyone got that.
Yes, sir.
Yeah. So as I said, that the major thing about this current session is that it is an interaction based session. So I would like everyone to ask questions at every turn, talk amongst themselves, at least use the hands up emojis or anything you get to interact with me and understand the course better. So to start off, let me first introduce myself. My name is Anuvab Chatterjee. I'm a Prime Minister Research Fellow and PhD scholar under the care of Professor Jonaki Shane at the Brain Developmental Biology Lab at the Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. As part of these sessions are being conducted as part of the PMRF deliverables, and we will have weekly sessions every day at Sunday from 4 to 5, and I will encourage you to join as many sessions as you can. But if you can't join one of the session for any due problems, that is also not a problem at all, because all of these sessions are recorded and will be uploaded to the my YouTube channel, in which you can go back and go across all the videos. So we are on board with this. Yeah. Hello. I guess since no one is, at least some emoji will do. Yeah. Like a hands up gesture that you are following what I'm saying.
Yes, sir.
Yeah. Thank you, Abdul. Yes. Abhina Parveen, yes. Srividya, Deepak. So to start off, let me introduce how we will do the sessions. First, I will show you a question from the previous years. We can have a vote in the comment section about which question and which MCQ is the correct one. We'll go through the content and the understanding behind the question in a couple of slides, and then we will answer the question. So if everything is okay, then we will start. This is the week 1 lecture, so I hope that everyone has already gone through, because already the week 2 lectures are live. But even if you have not gone through, I would say that week 1 lectures, if you have only gone through it momentarily, even if that is the case, you will still be able to answer the questions more or less once we have gone through the concept slides. So the first question is that we will tackle is viviparity is observed in, and the four options are: A, all mammals; B, placental mammals; C, all vertebrates; and D, mammals and amphibians. So I would like everyone to at least guess the answer in the comment section. And let me look at the comment section. So Abdul says V. Sri Vidya is also with B, placental mammals. Ahmed, Deepak, Abina Parveen. Is that Mrinalini Prusti? Mrinalini? How do you pronounce your name?
Sir, it is pronounced as Mrinalini.
Mrinali.
Mrinalini.
Mrinalini. Okay.
Yes.
So you also say B, right?
Yes, sir.
Yes, okay. Ujjwal is also going with B. So let us go through the concept slide of this question very quickly, because it was so here we have a table that differentiates between oviparity, ovoviviparity, and viviparity. The words ovo itself means egg, and ovoparity is the trait of laying eggs. Viviparity, on the other hand, is the trait in which birth is directly given to young ones. And in the middle of both of them lies ovoviviparity, in which the eggs are laid but they are kept inside the mother's body or womb till they are hatched. Oviparity is a general trait that is seen in many organisms such as birds, insects, reptiles, amphibians, and fish. Viviparity is seen in some reptiles, fish, amphibians, and of course mammals. Ovoviviparity is seen in some organisms such as reptiles and amphibians. So, can anyone cite an example of ovoviviparity? Oviparity is easy. It can be any birds, avians, chickens, anything. Viviparity is also easy. It is most mammals. And how about an example? Yeah, sharks. Yes, correct. Exactly correct. Sharks are a very prime example of ovoviviparity. So the type of fertilization that is observed in oviparity can be internal or external. In viviparity, it is internal, and in ovoviviparity, it is also internal. Next, about laying eggs. In oviparity, obviously eggs are laid. In ovoviviparity, also eggs are laid. In viviparity, eggs are not laid. Nutrients. This is actually the major distinction between the three. In oviparity, the nutrients come from the egg yolk. In viviparity, it comes from the mother via the placenta. And in case of ovoviviparity, it also comes from the egg yolk. Placental connection, obviously there is no connection in oviparity, no connection is in ovoviviparity, and a placenta connects the mother and the embryo in case of viviparity. And finally, the embryo develops outside the body in oviparity, inside the mother's body in viviparity, and finally inside the mother's body in ovoviviparity. Yeah, I would guess that is enough. Any questions from this? I guess not.
Sir, can you please explain how the growth and development takes place in oviviparous animals?
Oviviparity meaning the middle category. So in ovoviviparity, the eggs are essentially, it is more or less similar to oviparity, just that the eggs remain inside the mother's womb. There is no connection between the mother and the egg or the embryo, so the nutrients are already delivered inside the egg. It is, you can imagine it as if the chicken, instead of laying the egg, it is still inside the mother's body. That is what ovoviviparity is. Yeah, there was a question, I guess. What about scorpions? Okay, let me check. So, can you elaborate the question, please? Abdul, I think, asked the question. Would you like to elaborate on the question, Abdul? Hello? I'm audible, right? Hello?
Yes, sir.
Yeah, at least I'm audible. So, Abdul, are you there? I guess not. So, yeah. Yes, sir. So, I think it is overweight parity in case of scorpions. That is something that I would have to look into because I do not know exactly what kind of embryos develop in case of scorpions. So I will look into it, and I will. It is probably overweight parity as well as you are saying, but I will look into it and see. Okay, Abdul, are you there? Yes. So for our current question, the answer is obvious then. That the in case of viviparity, it will only occur in placental mammals. So why was all mammals not the answer? Can anyone tell me that?
So all mammals are not present in viviparity because some mammals lay eggs. Like there are exceptions.
So can platypus is a prime example of a mammal that lays eggs. So it won't be all mammals, but it will be placental mammals. Okay, so if that is okay, then we can go forward, right? Let me do this first. There is this is I think better. My screen is still visible, right?
Yes, sir.
Yeah. Now I can at least see who is talking at least. So we have been joined by Mary Aradhna. Okay, so let us move on. So the next question is one of the it is the crux of the whole. It is the crux of the whole course. Like it depends on one of the most important topics on the course, which is what is a blastocyst or what is a blastomere, or how are the stages defined in the whole development of an embryo, is what we will look into it in the next question. So, what is a blastocyst? The four options are: it is a stage of the embryo that has completed cleavage. It is the yolk-filled cavity in the amphibian embryo. It is the fluid-filled cavity formed by gastrulation in any vertebrate embryo, or it is the fluid-filled cavity formed at the blastula stage of the amphibian embryo. So I would again request everyone to at least try answering the question. You can just type whatever you like in the comment section. So we have D as one of the major answers. We also have A, stage of the embryo that has completed cleavage. Yes, I would also like everyone else to also try as well. D. Anyone else wants to try? So we have a tie, it seems, between A and D. So part of the meeting is saying that blastocyst is the stage of the embryo that has completed cleavage, and another half is saying D, it is fluid-filled cavity formed at the blastula stage of the amphibian embryo. So I think this is a tie yet. So let us go into the concept slides, and I hope that will clear us out. So this is a video. I would very much like to know if you are able to hear the sound of the video or not. It is a YouTube video. The link is also present.
We're going to talk about early embryogenesis. Say you're an egg cell.
Is this audible?
Yes, sir.
Yeah. So we are going to play the video, and please, I would like you to look clearly at the video itself because it is very important.
We're going to talk about early embryogenesis. Say you're an egg cell, and you have this nice thick outer glycoprotein coat called the zona pellucida, and you've got your plasma membrane just inside of that. And a sperm has made its way through the zona pellucida and managed to get in through your plasma membrane and merged its genetic material with yours. You're now called a zygote, and you'd like to go on to form an embryo, but not much is going to happen when you're stuck as a single cell. And so what you've got to do is divide into multiple cells, and you've got to do it fast. In fact, you've got to do it so fast that you don't have time to grow. So you actually just split into two cells, and this process of splitting without growth is called cleavage. And you do this a number of times, dividing from 2 cells to 4, and from 4 cells to 8, all the while staying within the zona pellucida. So you've gone from 16 cells to 32 cells, and at this point you look different enough that somebody decides to give you a new name. So instead of being called a zygote, you're called a morula. And morula is just a Greek word that means mulberry, and you actually do look a little bit like a mulberry. So here's a picture of a mulberry from my front yard to prove it. This one's not quite ripe. So you're finished with the cleavage stage of early embryogenesis. You've gone from 1 cell and just divided without growing at all into 2, 4, 8, 16, and then 32 cells. And now more interesting things start to happen. So you're still stuck within that zona pellucida. So we'll draw it in here. And I'm not going to keep track of the number of cells anymore, I'm just going to draw them in. But what you'll notice is I'm drawing them in quite compacted. I'm drawing them all right in the middle of the structure here. And they do look like they're a little bit tighter together. And this is a process that's called compaction. The different cells within the morula start to get closer and closer together. And in fact, the cells start to get a little bit different from each other too. And you notice that these cells on the outside are a little bit different. I'll draw them in here, slightly different color. And we can tell them apart, and this process of being able to tell cells apart as they become different things is called differentiation. So here we have 2 separate populations of cells. This one on the outside, we'll call them trophoblasts, and this mass of cells on the inside, we'll call embryoblasts. And so you're going to continue with your process. We'll move on to the next stage, and we'll draw our zona pellucida here, and we'll draw all of our trophoblasts along the outside here. Another interesting thing is happening to those cells in the middle. They're starting to clump even more. In fact, they clump so much that they all cluster at one end, leaving a little cavity on the other end. So here you can see your trophoblasts and your embryoblasts, this mass of cells in the middle. Some people actually call that the inner cell mass, and it's left you with this cavity, and that cavity is called a blastocoele. Now, again, you're starting to look a little bit more different than you have in the past, so we're going to give you a new name. This structure that you've turned into with a blastocoele and an outer cell ring of trophoblasts is called a blastocyst. In fact, we name this whole process after you, and after cleavage, the process is called blastulation. So also about this stage in blastulation, your zona pellucida starts to disintegrate away. Here I'm going to draw little bites being taken out of it here as it falls away and disintegrates. And that's going to be important later on because you can't be stuck in this thing forever. And the next step in blastulation, you actually just lose it completely. So now we'll draw our trophoblasts completely naked without a zona pellucida. But more interesting things are happening to you. Your inner cell mass of embryoblasts is starting to look a little bit different. So you still have this rim on the outside here up at the end of your...
So I would like just to add one thing that this particular stage of the blastocyst is very important, and this
blastocyst and
is very important and is very related to the question. I hope that the question is at least answered at this moment. Is there any question regarding the video till here? Because after this we move on to other stages, advanced stages of cleavage and development. Does anyone have a question till here?
Sir, I have a question that
Yeah.
So how the blastocyst, like how the process of embryogenesis occurs, as I have some of doubt in this process.
Yeah, so one of the major things here is that we are not looking at embryogenesis in very detail now. We'll look into it in very detail in further down the course, when we actually look into amphibian embryogenesis and how a frog egg develops. For now, this is a more or less an introductory video, but if you have any question that you want to ask now, you can very well go ahead and ask. Like in this video, there is not much that is defined about very crucial stages of how gastrulation occurs or how the blastocyst is formed. That is something that we'll deal later down the course. But if you have anything else that you want to ask, you can go ahead and ask. Yeah, Ahmed?
Yes, sir.
Yeah, do you want to, what was your question? Go ahead.
I just have a doubt, like how the blastocyst is, it has divided like the process.
Like how the process of blastocyst formation occurs is what you are asking?
Yes.
Yeah.
Yes.
If we just go back a bit in the video itself, you will understand that once the two cell layers of trophoblast or embryoblast divide, now the inner cell mass starts clumping together towards one end, so that one part of the embryo starts becoming empty. This is known as compaction. So the cells, after they have gone from the trophoblast to the embryoblast stage, there is various cell signaling that is going on, and this particular cell signaling goes through a molecule known as hippo, and depending on if it is at the edge of the embryo or towards the middle and the cells that are around it, it will understand that it has to undergo compaction or if it has to remain as trophoblast. Once it has gotten the signal, it now starts compacting, and you just have the embryoblast starts compacting into the inner cell mass, whereas the trophoblasts remain where they are, forming the outer lining of the embryo. Okay, Emma?
Yes, sir.
Yeah. So we move forward with the video itself. Anyone else has any questions regarding this? I guess not.
Can't be stuck in this thing forever. Are trophoblasts completely naked without a zona pellucida? But more interesting things are happening to you. Your inner cell mass of embryoblasts is starting to look a little bit different. So you still have this rim on the outside here up at the end of your blastocyst, and you still have this mass here in the middle. But you've developed another cavity, and this cavity is called the amniotic cavity. And also, your inner cell mass of embryoblast has started to differentiate more. And now it's got this layer on the bottom of it here, and the cells in this layer are called hypoblasts, while the cells in the layer just above it are called epiblasts. Now at this stage, we're pretty much completely free of our zona pellucida, and that's going to be really important for implantation. But that's a discussion for another time. We're going to focus on this ball of cells here. And I really want to stress that this is a ball of cells. It's not flat like we've got it drawn here on the computer screen. It's three-dimensional. It's spherical in nature. A spherical melon. So think of it like a melon. Here I'm going to draw a spherical melon. So now I want you to picture taking a big knife or a machete or something and just slicing the top off the melon like this. Here we'll erase the top part of our melon here, and we're left with a flat surface. So now that you've got a melon with a flat surface, picture yourself taking a pancake and just putting it right on top of this flat surface like that. And that's basically what we've got here with our blastocyst and this forming layer of epiblasts and hypoblasts. It's really a pancake. It's not two-dimensional here. It's a three-dimensional disc of tissue. So we're going to draw this again, but this time without the outer sphere of trophoblasts. But we're going to draw our pancake of epiblasts and hypoblasts. And what you can see here is our pancake has got two layers. So in fact, this is a very important structure in embryology called the bilaminar disc. And we're going to get another look at the bilaminar disc here. So I'll show you. We're actually going to draw a little plane through the disc here. And if we take a cut view through this plane that I've drawn, it looks a little bit like this. So this is still our bilaminar disc, but now we're looking at a slice through the bilaminar disc, as opposed to here we're looking at the entire bilaminar disc from the outside. So now we're looking at our pancake of our bilaminar disc here, and we notice we start to see something forming on the edge of it here. And that's something that looks like a little bit of streakiness that kind of splits our pancake right in two. You can think of it as pouring a little streak of syrup along the top of the pancake and dividing it into two halves. So here I'm going to draw it on the surface of the pancake, and then over here on our cut view, you can see that this little streak happens right about here in the middle as it reaches the plane of our image. And this streak of syrup on our pancake has got a name. It's actually called the primitive streak. And the formation of the primitive streak marks the beginning of the next stage of early embryogenesis, and that's called gastrulation. Now what that primitive streak actually is just the site where the cells in this epiblast layer of our bilaminar disc start to migrate. Here I'll draw the paths of the migrating cells. They heap themselves up right at the primitive streak, and then they start burrowing their way down to this bilaminar disc. And they go out into the hypoblast layer, and they just kind of go all over the place here. But it all happens from that primitive streak. So as the cells migrate out from the primitive streak, we can see that our structure has changed a little bit. Now instead of having two layers, one layer of epiblasts and one layer of hypoblasts, all of those migrating cells have now differentiated even further, and we're left with a layer of cells on the top, a middle layer of cells as they go out into the body of the bilaminar disc, and we still have a lower layer of cells down here. And so now you can see, instead of the bilaminar disc that we actually have a trilaminar disc. So we have three layers, 1, 2, and 3, and these are actually called our germ layers. So we have a single layer on top, and instead of epiblasts, this layer is now known as ectoderm, and our middle layer is mesoderm, and our lower layer is endoderm. And cells from these three layers go off to do very important things, and each layer forms its own specific structures. So that process of the formation of the three layers is called gastrulation. Once our three layers are formed, we can move on to the next step in embryogenesis, and the next step is called neurulation.
This much of the video will be enough. So we have dealt with a lot of concepts here and gone up to exactly gastrulation. And I've also looked into the three important germ layers. So, does anyone have any questions regarding this? Because this is very basic and very important regarding the
primitive streak have responsible for notochord formation.
So, primitive streak and notochord formations are two different, completely different things. Notochord is a transient embryonic structure that will allow the neural tube to form. Primitive streak occurs before the notochord forms. Srinash? Shians. Do you have anything else to add?
Yes, sir.
Yes, please. Go ahead. Like, you want to ask anything else? Is what I'm asking.
No, sir.
So, notochord, what notochord does is, see, when the ectoderm forms, so this is till gastrulation that we'll see. After this neurulation will occur, in which the neural tube will form and it will fold, giving rise to the spinal cord and the brain. But what happens is that the ectoderm also forms the layer of cells known as epidermis. That those are the pigment producing cells, and it is just below just the skin layer. So ectoderm also forms the skin and the spinal cord and the brain. But what exactly tells which part of the ectoderm to form the neural tube and not the skin? That is the notochord. Notochord is the signaling center which tells a particular part of the ectoderm that you have to form the spinal cord and the brain, that is the neural tube. And since notochord is present just at a particular place, every other part of the ectoderm forms the skin. Okay, Shiansh?
Okay, sir.
Yeah. Anyone else has anything to add?
Sir, can you just explain a bit from the bilaminar layer, bilaminar plane, and the primitive streak?
So the plane formation, yeah.
Yes.
I will need. I can do this. Wait. Can anyone if I do, wait, we can use Google Meet, some of the options we have in Google Meet. So if I do this, can everyone see the whiteboard that is forming? On. Is the whiteboard visible?
Yes, sir.
Yeah, very nice. So if it is visible, and if I start writing on it, is the writing visible? Should be this here. Yeah, is the writing visible?
Yes, sir.
Yes, very nice. So what is happening is, you have first, in order to understand how they are showing the bilaminar disc forming, is first you have to understand that firstly we had a spherical layer like this, which had an outer layer of trophoblasts and the inner cell mass. So this is not very nice. Instead, okay, this is not very nice. Instead I will show it right in the video itself. Is the PPT visible again?
Yes, sir.
Yes.
Yes,
Let
sir.
me go back. Yes.
We're going to talk
back
now, instead of having two layers, one layer of epiblasts and one layer of hypoblasts, disc from the outside. So now we're going to draw this again, but
so here you imagine that there is a ball present. The ball has this entire thing is a ball-like structure, where my mouse is visible. Yes. The entire thing is a ball-like structure. We take a knife and just chop off 1/4 of the ball. So here is the plane where that is chopped off. We have a clean circle. If we look at it from above, the cross section. If we look at it from above, we will see a circle, just a 2D circle, right? Deepak?
Yes, sir.
So you understand, right? If it is a ball, you chop off the upper quarter and look at it from above. You will see a circle.
Sir.
Then that particular circle has this formation, that it has two layers in it. And thus it is called the bilaminar disc. Now what we are doing is we are cutting it again across here, like this, like perpendicular to the plane of the screen, so that we can have another circle. So I will show it with my phone. My phone is visible, right?
Yes, sir. Yes.
So this was our disc. This was when we are looking from above. This is the disc. Then we cut along this area. Like this is here, we are cutting along here. So we get another layer for cross section forming here. And after we have cut this portion, we are looking from this side. You understand or no?
Yes, sir. To some extent, I did.
So you can also take it as layers of a cake. So this was a spherical cake to begin with. You cut off a large slice and then you are dividing it in pieces, so that you can now see the layers of the cake that has a cream layer, as a chocolate layer, as a vanilla layer. So here the layers of the cake has 2 layers, which then undergoes gastrulation. This is the bilaminar disc that we're talking about. So this will be much more clearer when I show a less detailed video, the next one. That will be easier to follow. So we can just go into that video if no one else has any questions regarding this one. Anyone else wants to ask anything? I guess not, so we move on. First, let me do this. Yeah, okay. So that video was a general description of how embryogenesis occurs in any embryogenesis occurs. This particular video is on chicken embryo development, which is a model organism for varied developmental biology experiments, and we will see how the chicken embryo develops in order to better understand the previous one. Again, please tell me that the audio is there. Audio is there, yeah?
Yes, sir.
Okay. So that was the entire representation of the growth of the chick from the embryo stage to finally the chick hatching. So previous video was from Khan Academy. This was from Poultry Hub, Australia. All the links are given in the videos. It's slides. Once you get them, you will be able to go back, refer to it. So does anyone have a question regarding this? So this was more of a less of a descriptive video and more of showing how exactly embryogenesis and development occurs and how amazing it can be. So any questions? I guess not. So Deepak, I think, asked about the yeah. So Deepak, you were asking about the the pancake formation. So when you look at least in this form, the cross section that you are looking at is actually what the previous video was talking about as the cross section you get after cutting the top of the inner cell mass. So this is the bilaminar disc, but this is the bilaminar disc after the primitive streak has occurred. So the primitive streak that they were talking about, it is this line. This is where the cells will start to go into under the various layers, and this is where the neural tube will form. So what is happening is, like if we cut the primitive streak at 90 degrees and then look at it from like look at the cross section, is when you will be able to see the trilaminar disc. Was that like at all helpful, Deepak, Shreyansh?
Sorry, sir, but it actually confused me a bit more, sir.
Very nice. So what is happening is that this is the bilaminar disc that is being formed after the upper layer. Like the author was talking about how the ball of cells was cut. I was also saying that ball of cells was cut on the top, and then you are looking from the top. So this is what the view will look like. Now,
Yes, I understood till that part.
So now what you are doing is that essentially you are cutting. So this particular line is in the y-axis. You are cutting it along the x-axis. You are taking a knife and cutting the whole embryo along the x-axis, and then picking it up like this and looking at it like this. So this was like here. Then you have made a cut like this, and then you are looking like this. Is that like?
Yes, sir, yes, a bit more clear, sir.
Nice. Okay, so anyone else has any questions regarding this? Otherwise, we will move on. So everyone else is clear? Okay. So we move on to the to answer the question. So after all this discussion, it should be clear that blastocyst is the fluid-filled cavity formed at the blastula stage of the amphibian embryo. This is, we are talking about the first video when the inner cell mass divided it from the trophoblast, and we have a layer or we have a hollow cavity under the inner cell mass, that is the blastocyst. Okay, we are everyone is clear on this? Yes.
In amphibian embryo, they are the lower region called gray crescent.
Okay, so amphibian embryo and the gray crescent that you are talking about is again something that we will look into at a very later stage, like in the course about and the gray crescent is not located in the bottom half. So in the amphibian embryo is divided into two parts. One is known as the animal pole, one is known as the vegetal. The gray crescent signifies the place dorsally opposite where the dorsal blastopole lip will form and where gastrulation will start. So the gray crescent and any stages that you are talking about, it is before gastrulation has started. Okay, so
yes,
you're understanding what I'm trying to say, right? That it is as the gray crescent is just after the sperm has entered the egg. The blastula stage, it has not yet reached the blastula stage.
Okay.
So this particular part, I think in this particular video that we are looking at, it is better, like more clear how exactly blastula is formed. Yeah.
Yeah.
So anything else anyone wants to add? So I would at least assume that the confusion between A and D is clear. The A people, where did they go? Mrinalini, Deepak. I think that we are now agree that it is D. Blastocyst is the fluid-filled cavity formed at the blastula stage of the amphibian embryo.
Yes, sir.
Yeah. So we go forward. Now this is something that we go forward from the previous extension of the previous discussion. So after what do the various germ layers give rise to? So this question is depending on that. The pancreatic cells are derived from neural crest cells, endoderm, paraxial mesoderm, and dorsal mesoderm. So neural crest cells are, I think if you have gone through the videos, you will recognize that these are cells that migrate out from the cells close to the neural tube that migrate out from there and can be pigment producing cells or primordial germ cells, also give rise to the formation of ear and other organs. Endoderm is one of the three layers that we have just seen. Mesoderm is the other layer that we have just seen. Paraxial and dorsal just means the positioning of it, that the where the mesoderm originated from. So, like, we can again go with the poll. I would like everyone to try and answer. Where exactly are pancreatic cells derived from? Neural crest, endoderm, paraxial mesoderm, or dorsal mesoderm? So you can just try, na? So endoderm is getting a huge, like everyone is like endoderm. No one has any other answers. Yeah. Anybody else wants to try? Mesometic. Brinda says mesometic. Yeah, mesoderm. Okay. So let us go to the. So our answers. We have a large population saying in endoderm, and Brinda is saying mesoderm. So let us look into it. What exactly is the answer to this? So this image is from Scott Gilbert, Developmental Biology. Here we can see after gastrulation is done, we have the three layers: ectoderm, mesoderm, endoderm, and another layer, another cells which are completely separated out from them, which are the germ cells. Ectoderm, here the lineages of the various germ layers are given. Here we have the ectoderm, in which forms the epidermis of the skin, the neurons and pigment producing cells from the neural crest. Mesoderm gives rise to the notochord, bone cells, tubules of the kidney, red blood cells, and facial muscles. And endoderm gives rise to the stomach, thyroid cells, and lung cells. So, to better understand this, it is that ectoderm gives rise to the epidermis and the neural tube. So, every neural structure, central nervous system oriented structure is ectoderm, as well as the neural crest, which form the pigments. Endoderm gives rise to our whole alimentary canal. So the whole from our mouth directly through the stomach, every one of the linings is formed from endoderm. But you have to understand that it is not just from the formed from the endoderm. There are mesoderm oriented components as well. But more or less, we can say that all the entire gut tube is formed from the endoderm, and all the muscles, organs, everything else is from the mesoderm. Blood cells are also formed from the mesoderm. Okay. So, in order to answer our question, pancreatic cells are all obviously formed from the endoderm. Okay, Brinda. Brinda was like it is mesodermic in origin, but it is no, it is endoderm. Okay.
Okay, sir.
Okay. So we move forward. So this is also a question related to what was exactly taught in the lectures, which is which of the following are with respect to the role of which of the following are correct with respect to the role of FGF4 during dog limb development. So one of the tips is that when the question has a square box like this, it is actually a multiple choice question. So just ticking one won't do. You have to tick at as many as possible that are correct. So we can again go forward with the choosing of the answers. So FGF4 during dog limb development. The options are: it promotes follicle cell cycle, promotes chondrocyte proliferation, promotes chondrocyte differentiation, and controls length of limbs. So do we have any answers coming forth? Brinda says length of limbs. Kavya says chondrocyte differentiation and length of limbs. C and D. Trishant is also like chondrocyte differentiation and length of the limbs. C and D. Abdul is saying B and D. Chondrocyte proliferation and length of limbs. Ujjwal is saying controls the length of limbs. So I should tell you, proliferation means when the cells are dividing and producing more cells. Differentiation is when a cell says, I will form a particular type of cells, a particular other type of cell. So chondrocytes are cells that are precursors to the cartilage or cartilage itself. It will differentiate into bone, so it will form bone. So promotes chondrocyte differentiation. Essentially, is it is asking the chondrocytes to form bone. So we will go forward and look into exactly what it is. So this is a bit complicated slide, but I would ask you to just be attentive throughout. During limb development itself, there are various signaling molecules that play very important roles. One of them is the FGF. The FGFs have there are various FGF present: FGF8, FGF4, FGF10. But in this particular question, we are looking at FGF4 or FGF8, which have comparable roles in limb development. So first, I would like you to look at the picture, which is titled B. During embryo development, how the limb does not essentially grow out of nothing. There is a small thickening of the near where the limb is going to form, and this forms a button-like structure. This button-like structure is known as the limb bud. At the distalmost end of the limb bud, where you can see a darkening right here, there is a signaling center, which is known as the apical ectodermal ridge. And the apical ectodermal ridge, or the AER, is what releases FGF, namely FGF8 in this case, right here. So we have FGF that is being released from this end of the distal end of the limb bud from the AER. So the question arises is that what does FGF do? What FGF does is it controls two things. Actually, it controls one thing which gives rise to the other. It asks the FGF, what exactly FGF4 asks is that. So let me go back. How does a limb exactly form? First, the limb, actually the cartilage is what forms. A cartilage analoga is first formed, which is essentially a cartilage framework, which then out of which parts of it differentiate into bone. That is how the entire limb is formed. What FGF4 does is it tells the various chondrocyte cells that you have divided enough proliferation is done, you have reached the length that you wanted to reach, and now it is time you go on to differentiate into bone and thus set essentially the length of the limb permanently. So we have examples of how exactly this works in an evolutionary way in the Dachshund dogs, in which selectable variation through mutations has given rise to such short legs. So this is something that was also discussed in the lecture itself. The various hunters needed dogs that are able to crawl into tunnels and catch badgers. So what they took is that they selectively bred dogs with shorter legs. What essentially happened is that these dogs have an extra copy of FGF4. What the extra copy of FGF4 does is it asks the dog's legs to differentiate faster, and thus bone formation occurs way before than it should, and thus the length of the limb is permanently short, that we can see right here. Thus what FGF4 does is it promotes chondrocyte differentiation and thus also controls the length of the limbs. So we also have long layer Dachshunds who have a truncated FGF5 protein, FGF5 gene, which allows longer hair growth. So this is an example of how a similar receptor or a similar signaling molecule can have varied effects. So FGF4 is controlling limb development, but FGF5 is giving rise to hair development. So with respect to our question, FGF4 obviously goes and promotes chondrocyte differentiation and controls the length of the limbs. Is it okay? Yeah.
Yes, sir.
Any question regarding this? So this is also something that we will discuss at length during the course in detail, during development of tetrapods. But I think it should be clear. So Abdul, you were saying that chondrocyte proliferation is controlled by FGF4, but it is not. It is actually chondrocyte differentiation. So next, we change gears and we will now look into mutations in particular genes that give rise to various defects. So the gene we are talking about here is KIT. The KIT gene is expressed in somites, neural crest cells that form the retinal cells, neural crest cells that form melanocytes, and primordial germ cells. So if you have gone through the lectures, you will know the answer to this. So I will ask everyone to try again what should be the answer to the this question. So these are again square boxes, so that means that these are again more than one correct answers. So anyone want to try? Hello. So Shriyansh is saying A, somites. Kavya is saying A and D. So primordial germ cells and somites. Kavya is saying A and D, somites and primordial germ cells. Brindalini is saying B, C and D, neural crest cells that form the retina, neural crest cells that form the melanocytes, and primordial germ cells. Okay, so let us look into the slide itself. Trishant is saying A and C. So let us look into the slide and then go back to the question. So birth defects. So why do we study birth defects? The important thing is that birth defects give us an insight into how human development itself occurs, and then we can study what the underlying mechanism of the birth defect was in various mice models or various animal models. This particular gene KIT, when it was mutated, it was seen. So the KIT gene is very important for three things. First, it is important for neural crest migration, which give rise to pigment forming cells, ear cells, and gut cells. It is also important for precursors of blood cells, and it is also important for primordial germ cells. So if the KIT gene is mutated, we'll have various phenotypes, which will include, obviously, the pigment formation will be hampered, as we can see right here in the picture itself, where the animal model in the mice and the human baby with the gene defect itself have almost similar phenotypes. Both of them will have problems with blood cells, so both of them will have anemia, and both of them will be sterile in nature. So if a particular syndrome, if the particular gene KIT is mutated, then we will have the following things happening. First is the neural crests that form melanocytes will be hampered, and the primordial germ cells will be hampered. So I think everyone was very much inclined on somites, because somites are something that is obviously from neural crest, but it is not something that is affected by KIT. Okay, everyone is fine with this?
Yes, sir.
Yeah. So this is another important topic, which is the Van Baer's principles. So this is also something that was directly looked into in the lectures, which is, according to the Van Baer's principles, which of the following are true? Adult features are not useful for classification. Habitat specific adaptations are generated during later stages of development. Larval forms of barnacle and shrimp are very distinct from the adult forms. Early embryos of distant phyla are morphologically more similar to each other than their adult forms. So these are circles, so that means that there is only one proper answer that we need here. So if everyone will try D. Early embryos of distant phyla are morphologically more similar to each other than their adult forms. So we have Brinda and Kavya saying D. Anyone else wants to try? Deepak is also saying D. Okay, so let us move on. and look into the what exactly are the von Bayer's principles. Shians is saying B and D. But Shians, the thing is that these particular questions, as I said, are circles. So this means that we only want one answer. These are the best answer type of questions. So we look into the von Bayer's principles. These are the 4 principles that von Bayer had observed, and these are very important regarding developmental biology, especially during the time that they were developed in. So what the principles say are: general features of a larger group appear earlier than specialized features of a smaller group, such that all vertebrate embryos have gill arches, notochords, spinal cords, and primitive kidneys. What it exactly means is that some particular general principle features that are important for all embryos develop earlier, and then various specialized features such as wings, claws, hoofs, and various other things develop later on. This is what essentially the four all the 4 points will tell us. The second is that less general characters develop from more general. That is, specialized characters are created once the general characters have already been established, such as the skin development in vertebrates. That is, various vertebrates have scales, many others have feathers, many others have hair on their skin, others have nothing. So what this means is that skin development occurs similarly in all vertebrates, but then the less general characters such as scales, hoofs, claws, these develop later on. Embryo of a given species, instead of passing through the adult stages of lower animals, departs more and more from them. So this is simple enough that when a chick embryo or a human embryo develops, it does not go through various stages of lower animals, such as it does not form a fish first, then a cow, and then the human. It obviously differentiates more and more from the embryo of the lower animals as far as it develops. Early embryos of higher animals is not like a lower animal, but its embryos are. So the embryos of all animals look very, very similar, is what exactly von Bayer's principles tell us. So when we go back to the question, it is obvious that the last answer, early embryos of distant phyla, are morphologically more similar to each other than the adult forms. That is, the embryos are very much more similar than the adult forms of the organisms, is the correct answer. So Shriyansh was saying B could be a correct answer, as that habitat specific adaptations are generated during the later stages of development. It is kind of correct that it is trying to say the same thing, that specialized features develop later during development, but the word habitat was not mentioned in von Bayer's principles. So the answer is the 4th one. Yes, Shriya, I guess that is okay, Shriya?
Okay.
Yeah. So which of the following are not consistent with preformation? That is, which of these options tell us that preformation was actually not correct? So the 4 options are: blood cells form before the heart development, left coiled snails produce right coiled progeny, inability in humans to regenerate a lost limb, or missing of certain structures when 1 or 2 blastomeres are removed from tunicates. Anyone wants to go ahead and try answer this? So this is actually a multiple choice question. Deepak is saying A and D. Blood cells form before heart development, and missing of certain structures when 1 or 2 blastomeres are removed in tunicates. Kavya is also saying DNA. Anyone else wants to try? Shriyansh is saying D. So let us go ahead and look into it, if anyone else does not want to try. Okay, so let us look into what exactly preformation is. So preformation was this theory which says that the entire generation of offspring occurs as a result of unfolding and growth of preformed parts. That is, the embryo already has a head, hands, tail, everything already preformed, just that this particular structures grow as the embryo develops, instead of it developing from scratch. This is what exactly preformation told us. But later this theory was discarded when development of early embryos were closely observed, where people could see actually neural tube forming from a flat surface of cells, or various cells forming before their organs are formed, or organs essentially forming from other cells, which tells us that preformation actually does not occur, but cells actually develop, and there is a process of morphogenesis that occurs that gives rise to the structure of the organs. This was also proved when examples of distinct phenotypic differences between generations were observed. What this means is that with preformation, it is obvious that a person will look like their previous generation. Like a man will look like similar to how his father looked, because preformation tells us that both of them were already preformed when they were when creator or God created every life. So more or less, various progenies will look similar. But this does not show us how exactly evolution occurs, because this will actually give rise to various changes or adaptation in various generations. Or another very important example that is often cited is that how left-coiled snails form right-coiled progenies. Because if preformation was correct, then all the snails should be right coiled, because all of them were formed at the same moment, so there should not be any changes. Okay, so this is exactly what countered preformation. So the options are that were correct, accepted, are blood cells form before the heart develop. So if blood cells form before the heart development, there is no preformation, because then heart needs to develop before it can be blood cells can be pumped around. And left coiled snails produce right coiled progenies, also not consistent with preformation. So D was one of the answers that was given very often. So missing of certain structures when one or two blastomeres are removed from tunicates. Essentially, this can also help preformation in a ways that one can say that the blastomeres were actually smaller forms of the limbs or whatever structures that give rise to the adult organism structures, and we are removing it. Actually, that part gets removed, and that is why structure is not being formed. Say the particular muscle of the tunicates is being formed from these blastomeres, but preformation, the people who support preformation will be like, those were not blastomeres, those were the muscles themselves in a smaller form, and they needed to develop. But since you removed it, that is why the adult does not have the structures. So it does not exactly counter preformation. Okay, is that fine?
Yes, sir.
You understood, right, what I am trying to say?
Yes, sir.
Yes, okay. So this is probably the last question that we are going to deal with. It is cardiomyocytes organizing into heart muscle is what process? Differentiation, cell migration, morphogenesis, or differentiation and cell migration? This is one answer correct kind of a question. So anyone wants to answer? Kavya says C, morphogenesis. Deepak also says C, morphogenesis. Shihan says D, differentiation and migration of cells. Anyone else wants to try? Rinda also says D, differentiation and migration of cells. Okay. Ahmed also says D. So let us look into the slide. One of the major things during development is called morphogenesis. Morphogenesis is actually a combined name for various biological processes which give rise to shape changes in cells, tissues, and organs. So large scale processes that actually form the various organs themselves. And morphogenesis is a huge thing that can be subdivided into various categories. So direction and number of cell divisions, this falls under morphogenesis. Cell shape changes, cell movement, cell growth, cell death, and changes in composition of cell membrane and secreted ECM components. So the entire formation of tubes from flat structures, formation of organs from various other smaller structures is what morphogenesis is. So any large scale organ formation always lies under morphogenesis. Thus, the answer is morphogenesis, but differentiation and migration is what you can call as falls under morphogenesis itself. So it is a higher umbrella of morphogenesis under which various processes such as differentiation, migration, cell death, cell division, everything else forms under. Okay, so like cardiomyocytes forming into heart muscle is obviously morphogenesis. Okay, everyone is fine with this?
Yes.
Yes, sir.
Okay. So this was everything I had for you today. So more or less, many of the questions that we have dealt with have references in the Introduction to Developmental Biology by Scott Gilbert, the 9th edition. So going through the book, you will find various of these images and all the data that is needed. Other citations that I have used, other references, all have the references given in the slides themselves. So I hope this particular session was helpful to you when you try and answer the assignments or go through the lecture videos themselves and try to understand them. You will find that when many of the concepts between the assignment questions from previous years and this year's is more or less same. So I hope that you will be able to answer correctly. And if you have any questions regarding this particular session itself, you can go ahead and ask it directly in the discussion forum that is present in the NPTEL course site. I hope that everyone at least has seen the discussion forum once. Yes, you know
Yes,
where
sir.
you know where the discussion forum is, right? It, yeah. So I am just telling you in case you don't. There is a ask a question section there in the coursework, like the course itself, the course tab. You will find the ask a question tab there, and you can just click on it, and that will open a discussion forum. And if you have any questions regarding the course, you can just type it down there. These sessions, so the session itself is over, but these sessions will continue weekly every Sunday from 4 to 5, in which I will discuss the weekly assignment questions from previous years and various concepts of the years, and you can ask me various questions about them themselves. I have lost my connection. Am I audible?
Yes, sir.
Yeah. So what I was telling is that these weekly sessions will continue till the course ends. So any questions you have regarding the course or regarding the various assignment questions, you can just ask me, or you can go ahead and put them down in the discussion forum. And this is the my YouTube channel link that is given right here. So all these sessions are recorded and this will be uploaded to my YouTube channel. And if you have any questions, you can just go ahead and rewatch the videos. If you are missing any sessions, you can go ahead and rewatch the videos in order to better understand everything that was discussed. Okay, so this was everything I had for today. Anyone has any questions?
So I have one regarding to the course. As far as I am aware, there are live sessions only of the doubt classes.
Yeah.
Are there live sessions of the course that is being taught as well?
No, as there are no live sessions of the course itself, but regarding that also you can directly ask in the forums, or you can also ask me in these sessions as well. Like if you have any questions regarding the weekly material, because I think that is also under the this particular session as well. So you can just go ahead and ask me anything regarding the coursework that is going on, the videos themselves.
So, sir, if I'm getting it right, we as we are getting messages of the videos of this week is uploaded at this link, so we are supposed to watch that only, right?
Those are any these sessions that we are doing here are additional sessions. These are completely up to you if you want to even appear for these sessions at all. But the important part of the session, the NPTEL course is on the site itself. The videos that are live videos that are uploaded, which has Professor Subramaniam who is teaching the course. These sessions are for your help themselves in order to better understand and help you to deal with the assignment questions, and also provide a live interaction between the NPTEL course team and everyone else, because more or less every NPTEL course are live are lecture videos themselves, and there is not many much human interaction in between them. So these are the sessions that will bridge the gap in between them, but these are additional completely. You can even if you want, you can completely go ahead with the course without even joining any sessions, but I would very much encourage you that you join and discuss everything in the course and the various assignment problems, that it will end up helping you itself.
Okay, sir. Thank you, sir.
Yes, anyone else has anything to add? So if not, then I would say thank you for joining the session, and I will look forward to meeting everyone next week. Thank you.